This is Dr. LeBlanc of the Department of Biology of Merrimack College. This unit focuses on the organization and packaging of DNA in prokaryotes. And if you remember, prokaryotes are single-celled organisms that do not have nuclei. The learning goals for this unit are to understand the organization of functional sites, which are the origin of replication, individual genes, repetitive sequences, and intergenic regions on prokaryotic chromosomes, to understand prokaryotic DNA packaging hierarchy, and to understand the action of enzymes that are involved in packaging that are targets of antibiotics. This is an electron micrograph, a pseudo-colored picture that shows a single bacteria cell right here with its single chromosome splayed out all over the surrounding area. This DNA is approximately 1 millimeter long. The bacteria is approximately 1 micron long. So there's 1,000-fold packaging that has to go on in order to get this DNA packaged nice and neatly inside the bacteria cell. Prokaryotic genomes are typically circular in nature. They are a single molecule. There's not 2 copies, but 1 copy per cell. Bacterial chromosomes have an origin of replication, a sequence of nucleotides to which the replication machinery attaches when you want to make a new copy of this chromosome before cell division. On the other side, there are termination sequences for DNA replication. There are genes, these are genes here, intergenic regions, and some repetitive sequence elements that are probably vestiges of past viral infections at some point during evolution. Here is a bacteria cell, here pseudo-colored again, to show how the DNA is compacted into a structure called a nucleoid. This is not a nucleus, it is just a region of the bacteria cell where the DNA is concentrated. This bacteria cell is about to divide. You can see a slight puckering right here. The cell is going to split in 2. And so each new cell will have its own nucleoid, which contains 1 copy of the bacterial chromosome. So here's our DNA, and we need to compact it so that it fits inside a bacteria cell nice and neatly, not tangled, so that it doesn't break and cause mutations. To fit into the bacteria cell, like I said before, it has to be compacted 1,000-fold, and proteins will bind to the DNA at specific sites and organize it into loops. Here are the proteins that have bound, and like a purse string, they've all gathered together in the center and has created loop domains in the new bacterially compacted chromosome. The loop structure compacts at about 10-fold. So the number of loops varies according to the size of the chromosome and the species. For example, E. coli has about 50 to 100 loops with about 40 to 80,000 base pairs of DNA in each. Now, 10-fold process compaction is not enough. We need to compact it further. So an enzyme called gyrase or topoisomerase introduces supercoiling into each of the loops to compact it another 10-fold, another 100-fold, so we have enough compactions for the DNA to fit inside the bacteria cell. Supercoiling is important to compact the bacterial chromosome, but cells also use supercoiling to store some energy so that we can separate the strands of DNA for some particular and very important functions in the life cycle of a DNA inside a bacteria cell. Okay, so here's a strand of DNA right here. There are plates that tether the ends of the chromosome or the ends of a loop domain. They prevent the DNA ends from rotating freely. So a normal non-supercoiled binding of the DNA would produce 10 bases per turn. If you underwind the DNA, you end up with fewer turns. You could have 12.5 base pairs per turn. This is not a stable structure. What happens instead is that part of the DNA loops out to restore that 10 base per turn, plus you get one negative supercoiling. You can also overwind and get more turns to produce approximately 8.3 base pairs per turn. This is also not a stable structure, and it's accommodated by creating a positive supercoil. So these two structures here and here are not compatible with living cells. They don't occur in living cells. However, the conformations with the negative supercoils or the positive supercoils are found in living cells, and they're called topoisomers of each other, and they are produced by the action of gyrases and topoisomerases. So DNA supercoiling can affect chromosome function. This occurs both in prokaryotes and in eukaryotes. Here's our bacterial chromosome with a negative supercoiling area right here. Negative supercoiling compacts the chromosome, and it creates some tension that might be released by strand separation. That's what this is showing right here, strand separation. We've broken the hydrogen bonds between the two strands of DNA, and now we have an open region. Now, we talked about the fact that DNA is a very functional molecule, and it has all these lovely hydrogen bonds holding the two strands together. Why on earth would we want to separate the two strands? Well, there are two processes in the cell that are critical to have access to individual strands of the double-stranded DNA molecule. One is replication. We need to use one of the strands as template and produce another strand. And the other is transcription, where we want to make an RNA copy of the DNA. So replication and transcription require this bubble to form in the DNA molecule, and topoisomerases and gyrases play a critical role in establishing the negative supercoiling that will result in strand separation under the proper circumstances. This is the action of an enzyme known as gyrase. Gyrase is a topoisomerase. There are two types of topoisomerases. Topoisomerase 1. Topos 1 break one of strand of the double helix, and topoisomerase 2 break both strands of the double helix. So gyrase is a topoisomerase 2 because it breaks both strands of the double helix. We're going to look at each of these panels individually. So gyrase has domain structure. It has the A domain in the lower structure and the B domain in the upper structure here. So when a DNA molecule binds, it gets looped through the two A subunits. Notice the orientation of this blue strand of DNA. It's going behind the first one and over this second A subunit. Okay, so it went behind and then over. Then it loops around and comes down the front. So we went behind this one, over this one, and then behind the B structure here. When this loop has formed around the topoisomerase, it's ready to be cut by the lower jaws. So the DNA is held in the lower jaws and it's cut right here, both strands of the DNA. If there is a strand here and a strand here, double-stranded DNA, both strands are cut. And here you can see the gap below here. Now, what's going to happen to this portion of the DNA strand that was over on top here? It's going to get pushed through and behind, so that now this portion of the DNA here, which was behind it before, is now going to be in front of it. And you can see that here. So this strand of DNA that was on top of this portion here is now behind it. This part is behind. Okay. And you can see the difference at the end of the process and the beginning of the process here. This strand of DNA that was on top is now behind. Okay, and you create these negative supercoilings in the bacterial chromosome. This is a very tricky operation. It's very critical that it is done absolutely perfectly, especially after the DNA is cut. You do not want to lose these ends. You can't let go of these ends because you have to reseal that phosphodiester bond exactly the right way without missing any nucleotides or adding any extra nucleotides in the process. So the ability of gyrase to introduce negative supercoils into the DNA is crucial for bacteria to survive. If this gyrase is inhibited or mutated in some way or knocked out by an antibiotic, the bacteria will die. It needs this to survive. So this has become a target for antibiotics that are pathogenic. Two main classes of drugs that inhibit gyrase are quinolones and coumarins. They do not inhibit eukaryotic topoisomerases, which is a good thing for us because humans are eukaryotes. So ciprofloxacin is an example of a quinolone that inhibits DNA gyrase. It's used in the treatment of anthrax and other bacterial infections. You may have known someone who had Cipro, or you may have had it yourself, not for anthrax, but for some other disease. So here are some of the structures of two different coumarins, novobiacin and chlorobiacin. Okay, for those of you who have already taken biochemistry, this is a competitive inhibitor, and it binds to the gyrase enzyme at a site that overlaps the ATP binding site. So it competes for ATP binding with DNA gyrase. If you'd like some more animated versions of this story, you can look at these two videos. In summary, we've learned that the organization of functional sites along bacterial chromosomes includes things like the origin of replication, genes, intergenic regions, and repetitive DNA sequences. We learned that bacterial chromosomes are organized in nucleoids. We learned about the structure of hierarchy of compaction of the bacterial chromosomes, loops, and then supercoiling. We understood the mechanism of gyrase, and we also learned that gyrase can be a target of antibiotics.